Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
A globally rare variant of the growth hormone receptor also seen in Denisovans and Neanderthals may have helped our ancestors survive periods without food. A new study published on September 24 in Science Advances claims. Reconstructing the evolutionary history of a particular variant of the growth hormone receptor gene, GHR, variant. The so-called GH-ROD3 variant, which is defined by a deletion of the gene's third exon. Researchers have found that its frequency declined sharply around 40,000 years ago. Follow-up experiments in mice pointed to a potential explanation for why. The deletion appeared to limit male animal size when fed a calorie-restricted diet. This sort of growth limitation could help males survive lean times but limit their reproductive success in times of plenty. This paper is exciting because it illustrates the power of combining evolutionary analyses of modern and ancient genomes with in-depth molecular characterization of the effects of genetic variants, writes Tony Capra, a specialist in evolutionary and computational genomics at the University of Cal. I believe that the context-dependent complexity revealed here for the effects of the GH-ROD3 variant, in terms of sex and environment, are likely common. We just haven't been able to explore the molecular effects of other variants with evidence of recent selection in such detail. In previous work, Omer Gokcuman, an evolutionary biologist at the University at Buffalo, and his colleagues scanned Neanderthal, Denisovan, and ancient and modern Homo sap. One such variant that they identified was GH-ROD3, so for the new study, they dug deeper into its evolutionary history and potential function, to get at why it might have persisted for so long. Initially, the researchers hypothesized that the variant may have been maintained by a phenomenon called the heterozygous advantage, where individuals that carry both versions of a gene, are heterozygous, have an advantage over people who carry duplicates of one version, are homozygous. Instead of being maintained at stable levels throughout human history, GH-ROD3 emerged as the dominant growth hormone receptor allele in ancient humans roughly 1 million to 2 million years ago, and it stayed that way for a very long time, until 30,000 to 40,000 years ago, when it rapidly declined in. In the literature, GH-ROD3 is associated with smaller birth size, early onset of puberty, and increased longevity. Traits that Gokcuman observed are also associated with a starvation-related phenomenon known as catch-up growth, where growth later in life compensates for smaller birth weight and size. The team decided to look closer at GH-ROD3's potential role in starvation resilience by examining the frequency of the deletion among children in Malawi who had survived malnutrition. In other words, the deletion seems to be protective against that particular metabolic syndrome, says Gokcuman. To further interrogate the link between malnutrition's effects and GH-ROD3, the team used CRISPR-Cas9 to knock out the third exon from the mouse version of the gene, generating a murine model of the gene variant. That revealed a sex-specific difference. When male mice carrying the deletion were fed a calorie-restricted diet, they ended up weighing the same as their female counterparts, while in wild-type mice, male mice were always larger than females. The deletion had no effect on female mouse weight. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.